Hi everyone, so we're going to take a closer look at the chemistry behind nucleic acids. This is scary for biologists, much better for biochemists and chemists of the such, but it is super important as we get down the line talking about how we basically activate DNA genes, why RNA's nucleotide is so dynamic, and basically kind of how they relate to proteins, because we're going to kind of contrast them with those. And this is kind of the fun leading question that I always like to start with, which is where do replicating nucleotides lie in the kind of the border of what's alive and what's not? We know objectively the border of what a living thing is a cell, but that's contained in a plasma membrane, things like that. So keep that in mind as we keep looking at these nucleotides, as you'll kind of be able to see that they can add to themselves, they can be dynamic. So in your mind, kind of see and justify where you define, where that emergence happens. And that is the word used to define basically the transition from non-living components to a living thing. So like any biologist, chemistry terrifies a lot of us, right? Orgo, oh no. You'll see that a lot of what you learned in Orgo will at least help you feel more comfortable with the dynamic nature of nucleotides and amino acids as we go. So nucleic acids, there are two main ones, obviously, DNA and RNA, okay? We know that DNA is going to link up and form two strands like this, very rough looking like that. We know that RNA is just going to form one strand, okay? Both of them have this big phosphate right here, and both are going to connect up here like this, and they're going to they're going to connect to the next length of DNA. So on this poorly drawn strand right here, they're connecting through the phosphate backbone up here. One thing to note is that the phosphate backbone of DNA and RNA is highly negative. It's also huge. This chemistry, this bubble right here doesn't show how big phosphate actually is. It's massive. So it's this big protrusion, and it has these massive negative charges here. And that's something similar with both. Okay. Next is that we do have a little base right here for each one. And the bases are different. There's purines, pyrimidines. That's something that you can kind of read about and kind of obviously coming into this class, you should kind of know and understand. For the most part, they're going to be the same, except in DNA you have thymines, and over here in RNA, instead you have uracil, and highlighted in blue is the main difference, is you lose this carbon with uracil. The primary difference here is that with DNA, you only have one reactive OH group, and those of you that are chemists know how reactive OH groups are. Electrons can basically attack from this position. With RNA, you have two of these groups. This is going to add a lot of differences between RNA and DNA. One of which is that RNA has all of these OH groups out here, this extra one. That means it's going to be reacting, and other things are going to try and degrade it. They are going to come after this. That's why RNA usually is typically a lot more unstable than DNA. Because remember too, DNA, like we set up here, this thymine right here is going to attach to an A. I'm not going to draw it. Well, I can draw a little A. Yep. There we go. Meanwhile, this uracil is not going to connect strictly with anything. We'll see where that can actually change up in this class. Okay, so I don't think I missed anything. There we go. Yes, okay. So like I was talking about here, I probably should have just gone to this slide. The main role of this phosphate group is to connect to the adjacent nucleotide, as you can see here. So see here, this is why we call it that 3' terminus, and I know I just got rid of that. This OH is going to react with the next phosphate. See how this phosphate of this nucleotide is actually attached to previously what was a full OH of this nucleotide, so on and so forth, right? And so ultimately at the 5', that's why you have, this big phosphate. That's also why everything, in DNA replication, and again, you've seen that in intro, you've seen that in, 201 or your typical genetic cell class. That's why everything is always added in this direction. It's because you have this nice reactive OH right here. That's the main reason. This is reactive. Up here is not reactive as far as adding nucleotides. Okay, so when we get into that, that's the reason why we go from 5' to 3'. Okay? As you can see with RNA, see in red here, you have this huge reactive OH group. So it's not necessarily large in size the same way the phosphate is, but these are massively reactive, okay? And these nucleotides are not going to attach to a double strand. They're not going to make a helix like over here. Each one of these is going to make a nice interlocked helix. These are free. That means that all these groups right here are free to bind. We'll see where they go. These are also free to bind, and so is this OH down here, okay? Okay, so keeping those structures in mind, now you can kind of take a look at a protein. So with an amino acid subunit right here, you have that alpha carbon that is going to connect to four specific things, I guess if you count the hydrogen. They're always going to be connected to a small hydrogen, they're always going to have a small amino group, they're always going to have a small carboxyl group, and then this is where you get your different amino acids, is that massive R group. So we'll cover this more in the next thing where we kind of cover strictly proteins and amino acids. But notice that these have a much more dynamic shape when it comes to forming a single-stranded molecule. Also notice how many more reactive R groups or even the carboxyl groups that you have sticking out everywhere. Proteins have a lot more possibilities. And remember too, with these R groups, there's 20 different versions of these. So if you take a look back up here, there's only 4 types of nucleotides on DNA. There's only 4 types of nucleotides, pretty much on RNA. They don't have the same diversity as something that has 20 possibilities at this, this purple section right here, okay? You'll notice that it follows a similar pattern where, the amino group is going to connect to the carbon that contains the carboxyl group of the adjacent amino acid residue, and so on and so forth, and you can form a polypeptide chain. So there are similarities given that, each one kind of has a backbone, each one has, if you want to call it a variable group, that's fair. But the only variation you'll see with nucleic acids is purines like A and G, pure like silver, A and G, and then pyrimidines like thymine, cytosine, and uracil. So this size difference is going to make a difference, and there's some differences in these kind of the chemicals there that we can see. Okay. So first things first, when we see DNA, this is something I was trying to note before. Oh, I didn't want that. Okay, there we go. DNA is going to be interlocked right here. That means that these nucleotides in here, so all this, those are safe. They are not going to interact with anything. Meanwhile, with an RNA, remember we saw tRNA, how it forms this amazing 3D shape? Okay, let's just readjust. Sorry about that. With these nucleotides are interacting basically in whatever way is most chemically favorable, okay? So the specific combination of nucleotides is going to favor certain nucleotides binding with other nucleotides. Maybe some nucleotides form, 3 bonds at a time. Maybe some are attracted to different OH groups that are sticking out of, this RNA strand. You can find structures like this so much, and they do have some function here. So this slide is mainly here to show you that RNA has a lot more dynamic 3D natures than DNA does. DNA is likely going to form something like this. We will get into kind of the superstructures, the wild structures that DNA can do. But RNA has fewer limits. So remember how we talked about exons, how you have, the big strand of RNA, but you have to cut out, this intron or this intron and this intron, let's say. Some introns actually have sites, and they form a 3D shape that goes and it forms basically its own loop, and it cuts itself out. That's what you see right here is basically, let's say this is the, there's an exon and then there's this big intron. Basically that the chemistry right here actually causes a chemical reaction that cuts out this loop right here and removes it. That's what these self-cleaving RNAs or these self-splicing introns are basically doing. They can interact with each other. They can form chemical reactions. Remember we saw this with that ribozyme? That's why RNA is so special, is that it can take on a 3D shape. Only proteins can do that usually. DNA is so stable that it rarely leaves this double helix. RNA does have some dynamic properties where it can actually go and do, like have an actual function, right? RNA can also be quickly changed by the composition of its nucleotides. When you change the small chemical groups on a nucleotide with DNA, usually it's kind of bad. It usually means that, it's not going to have that nice double helix binding, and it might actually end up in a mutation. If you change the RNA, nucleotides, and get rid of this here. You can make something less or more reactive, for example, and that can change the capabilities of these 3D sites. So this is kind of like a way that some cells can have an on and off switch deciding what their big 3D RNAs are going to do. Are they going to be on or are they going to be off? Okay. This is the first thing that we have as far as DNA modifications. And we'll see there are a lot of different modifications. We'll talk about protein ones, we'll talk about RNA, we'll talk about DNA. These are the primary things that you can look for when it comes to DNA modifications. So number one, you've probably been exposed to before. When you split DNA, for example, and then you start reforming, when you start reforming the DNA, see it's like going to do a new strand right there. It marks the parental strand by methylating it, okay? And you can do that on cytidines, you can do it on a couple others too. As you can see down here, it's usually cytosines that get methylated right here. Certain regions, when they see cytosines though, that can actually silence the expression of the DNA gene that is in that region. So if there's an active coding gene, but you put down a ton of methyl groups, that actually inhibits that transcription process, okay? The other neat thing about methylation is that it's actually going to prevent viral DNA from coming in and cutting itself in. So what we know about viruses, the basic thing is that they have, well, retroviruses in a lot of cases, there are plenty of viruses that have enzymes that basically cut the DNA and insert their own genetic material. They have a very hard time doing that if it's not natural looking DNA. Usually they would maybe need to cut right near a cytosine, or cytidine, sorry. And if it's methylated though, they can't cut because the viral enzymes are so specific they need to have that. But if they do get in, one way you can silence them is similar to right here. You can actually methylate a region. And so the cool thing about 4 right here is that our body has tons of relic viruses out there that are stuck in our genome from, trust me, millions of years ago that were infected, but our body's reaction was actually methylate that region that got incorporated, and it just stays silent. It's just there waiting. We'll read a quick paper on that coming up. Next thing is that some regions of DNA, if they're not methylated, they can actually be sensitive to like enzymatic cutting basically. So this is a very good way to protect those regions and basically say stop, do not cut this region, so you hypermethylate it and make sure it's good. Okay. So talking all about this virus stuff, this kind of brings up that leading question that we had, are they alive? So in your typical life dogma, you have something that has a DNA blueprint, RNA transition, functional proteins that are going to, have roles all over. All a virus is nucleic acid that can self-replicate, but it needs a host to do that. As you can tell, that can be in a eukaryotic cell, like a human cell, where it's going to insert its DNA or RNA. This is going to create more products that ultimately engineer and create lots more viruses themselves, right? And then you have more viruses, they go and infect more cells. That's how it works. The same things happen, similar things happen in bacteria and archaea. Sometimes viruses can actually pose as circular DNA plasmids, which is super interesting. And in a lot of cases, we'll meet these transposons. Sometimes the viral genes that are meant to, cut and paste and then express their genome, they kind of instead, some viruses really don't harm their host as much as they just bounce around. What I mean by that is the nucleic acids get inserted, but as you can see from these small arrows, they kind of just hop to a new region of the genome, and that's the point of them, that's it. We'll see that actually causes a very interesting set of roles and criteria, especially when we get into human genes that resemble viruses. But just know that basically the way that these things operate, they are evolving, not living nucleic acids, but they are moving and creating more of themselves. And sometimes they have roles that go way farther beyond disease, for example. And that's one thing we'll learn in this class is their role as nucleic acids. They're not just, they don't cause damage all the time. We'll see that there are human genes that are likely actually viruses that perform a lot of really important functions for our cells that we kind of co-opted. So we'll get to meet those later on. Okay, So we talked a little bit about RNA and DNA modifications. We will meet all of these as we go on, but protein modifications, well, we'll meet some of these as we go on. Protein modifications, there are tons. So like we said, if you have tons of those variable R groups and amino acids are just, easier to modify in general, you can add on tons of different stuff to different amino acids. As you can see here, here's a serine, here's an arginine, and each of these R groups is going to have a different characteristic set of what kind of modifications can you actually put on it. And that adds to that massive complexity. And to be honest, we don't always know what one of these is going to do. I would say that phosphorylation is the one we know the best about, because phosphorylation is usually basically it can serve as an on or off switch. So when you add this phosphorylation to a serine, this massive phosphate can cause a conformational change. And remember, it's negative too, so it's big and it's charged. This can cause conformational changes in proteins to turn them into an on state, where they fold into a new 3D shape that's actually now going to be active. It can also turn them off in a lot of cases, and they fold into a shape because of this phosphorylation that stops their dynamics, basically. Okay. This is pretty, I gotta be honest, this is pretty simple, but it's always good depending on where we're, if we are true biologists, we always need a good fresh reminder of where we're at. So one thing the reason I want to highlight these two bonds is that basically these are really massive, strong bonds, especially a covalent bond. What I want you to mainly know about this is that there are covalent bonds that are happening in our nucleic acids that we're going to talk about. There are ionic bonds that are going to happen in the chemistry that we're going to share. Know that what these two are just massively strong bonds. Breaking these bonds is difficult. And in something as reactive as a genome, you need to have bonds that can be broken at will to turn things on or off whenever you want. So a good example of a covalent bond is the phosphate backbone. But although they're very important, what keeps our genes and our DNA and our RNA and even proteins in some cases with some of their interactions, but mainly DNA and RNA, and mainly DNA to be honest, it's usually these really small bonds. It's usually small hydrogen bonds. So let's say right here in a protein, it's amino acids that are just barely interacting with polar or even small charged groups. So this positive arginine and these negative glutamic acids are sharing this weak charge right here. But in a lot of cases, it's even small ionic charges that are, or even, really faint things. So it's pretty rare that you see a dynamic molecule or something with a dynamic function that can, be turned on, off, high, low, to have that interaction be something that's breaking something strong like a covalent bond, okay? It's usually going to be something weaker, like small charge bonds like this. Okay, this is in the PDF. This is your basic organic chemistry. How many bonds are you usually going to see coming off one of these? Carbon, you don't spend enough time, true biologists don't spend enough time on carbon. You really can't get away with the diversity of life as we have it without something like this that can bond to 4 things. Silicone has 4 bonds too, but carbon is small and it can have nice strong bonds in each direction. Silicone is massive, so its bonds are like just tiny and they're not really going to hold on to anything. So that's why carbon is just like the indispensable molecule. It's really cool. And again, yes, we can form double bonds, single bonds. As you might well understand, a double bond is going to be harder to break than a single bond if we decide to even break a single bond. This is still really hard to break. And then my favorite, orbitals. So lovely. They are really important though. So in this tetrahedral right here, we can also see that depending, in a double bond right here, things go planar. But with single bonds, in a lot of cases with carbon, you usually have this nice tetrahedral formation that is really going to allow a lot of these molecules to go in different directions and have a ton of flexibility. That's the big thing to take away from this slide here. And again, I told you you'd see some orgo. The way that, polypeptides as we see right here, or nucleic acids, have the flexibility to, transfer energy or take on new shapes is because that the nature of having something, like a carboxyl group that can take on, an extra piece of charge, basically. Having something that can shift back and forth. I'm just going to add one right here too. Something that can basically have the ability to shift from charge to uncharge and kind of be in that dual state all the time, like a phosphate, for example. As we can see here, you can change where the negative charge is. That's what gives nucleic acid and the genetic material that we have so much power in a lot of cases, especially when we get deeper into the chemistry, is that the bonds and the charges are not rigid in a lot of cases. They are not set in stone. They're not something that our genome cannot figure out. But the most important thing is how these behave in water. And it's the first thing you see in intro bio. But again, like carbon, but in a different way, water is the indispensable molecule. There are very few things that are this small that carry with it positive and negative small charges here, so that not only can these, obviously more than millions of all these water molecules can all interact, positive touching negative, negatives touching positive, and it's a nice free flow. Something like methane, everything's balanced. They don't necessarily, cause any trouble, but they're not going to be able to bind to anything and basically make it happy. So if you have something like DNA, for example, remember that has that big negative phosphate, the water on these two is going to need to basically facilitate the negative phosphate being out and exposed, to the water in the aqueous environment of a living thing. So again, hydrophobic interactions also, we'll see, are going to be a key tenet, not just the hydrophilic ones. Should have mentioned that. Okay. This is basically, I can't remember if this is actually, oh shoot, should have checked this out. I can't remember if this is actually van der Waals or if this is just like kind of a polar reaction here. Sorry, this is what you want. But essentially there is a distance that is nicely maintained between two molecules that are like charged. And so I do believe this is van der Waals actually. And the way that orbitals work, you're going to have this positive and negative charge that's going to kind of come together just by proximity in a lot of cases. So that's something to keep in mind as we talk about these interactions. Now I'd say this slide is probably the most important, putting everything together that we've seen though. This is a DNA molecule. The outside and the orange right here, those are those massively negative phosphates. So I just talked about how outside here, this is all water. So all these negative phosphates are being surrounded by those nice positive poles of water. Inside though, the bases themselves, these are hydrophobic. These do not like interacting with water because they don't have the proper polar charges to do it. They are pretty nonpolar, if I'm going to be honest here, and they prefer to interact with each other instead. And what you're going to see is this is where the hydrogen bonds not only are forming, but the way that these are stacked. So I'm going to assume you know about the hydrogen bonds, that's why I put that there. The way that these are stacked on top of each other is basically it's a protective measure. Okay? The DNA, much like a Roman phalanx, is going to protect the one on top of it and to the bottom of it by pairing up with another one. So Roman phalanx is basically where you have, soldiers lined up with shields, and each one of them protects the soldier to their left. And so everybody's protecting the next person, and that's kind of what's happening here. Water can't get in because these are all stacked on top of each other, just like shields in that case. So you get this amazing, structure, this really, sadly a little bit rigid structure, but it's protected from the water in this case. And this is all facilitated by really weak hydrogen bonds. So between each of these nucleotides, that's where these hydrogen bonds are forming. And you've seen those before in your typical intro or your typical secondary level cellular genetics course. So again, why is this so important? These phosphates are linked covalently. See all these and the way the nucleic acids, these are hard links. These are hard. It is very difficult to break the backbone of one of these. You need energy. We'll meet enzymes that do it when they edit DNA or when they do stuff, but it's hard. But to open up a gene for transcription, to get these and to copy these like we saw with transcription, we need to open this up. We need to unzip DNA. And the real reason behind why these H bonds are so important is that you can unzip this very easily because these are just weak little H bonds. You don't have to break any, true chemical bonds in here. These are just hydrogen bonds. These are small polar bonds that, under the right circumstances, we can open up. And when you open this DNA helix up, you can copy it. That's where you get your RNA. RNA becomes protein, and you're off to the races. So regulating that on and off is key. And the key to making sure that you have a nice, unzippable DNA are these hydrophobic interactions. Anything that closes these interactions actually causes damage. Okay. So another way that H bonds are, obviously play just a huge role is think about the active site of an enzyme. And, we'll meet tons of enzymes along the way and they'll do all these fantastic things. That's why I will always like going into the details of the chemistry is that imagine this dark purple here is, kind of the interior of an enzyme. And here are all of its groups kind of sticking into this site right here, this active site. In this case, you have a lot of R groups that are perfectly positioned. And remember, we'll get to see proteins, how they can take on amazing 3D shapes that we have no way of predicting, in all honesty. They facilitate the perfect environment to not only interact with each other through the water, but interacting through their H bonds and their O's with the water's own bonds as well. This doesn't, yep, I knew that would make some terrible shape. So that's what an active site is. It's the perfect structure of all of these small amino acids. So I know that you remember we're outside of DNA again. So now we're talking about something different. This is what, when we talk about an active site and we do a little Pac-Man when we're showing you what what an enzyme is, these Pac-Man basically have chemical teeth, and that's what you're seeing in this slide. These things are going to be able to react with whatever comes into this spot. When something's not in this spot, when their target substrate isn't there, water occupies it. Okay. Now there are even, so I know I always butcher van der Waals like I did up there. But again, van der Waals are basically proximal contact binding and, millions or thousands of interactions. What an antibody is this big Y-shaped protein that your B cells make. And these basically variable areas on the tips here, they are going to bind with something called an antigen. An antigen is basically a piece of a pathogen. So this green stuff right here is a piece of, let's say, a virally infected cell that is showcasing, look, I have virus, or this is a piece of bacteria that is running around. And what your antibody has done is it's evolved to have these regions right here. These are amino acids. And it has evolved in a process that I think we'll get to go over because it's exciting and it's what I research. These variable regions have evolved so that this region right here, as I've highlighted, is full of amino acids, like we see here, that perfectly fit the contour and the chemistry of the antigen. And when you bring these together, it's a perfect fit, unlike what I just drew. And that fit is maintained and it's sticky, not only because of those H bonds, but because of the thousands of it's so perfectly structured, the thousands of van der Waals that basically it's like two hands like coming together and sticking, So in this case, this is the key in genetics and specifically molecular. It's not so much the massive, covalent, strong bonds that make this system so special and dynamic. It's usually these, it's thousands of these weak, small ones, these tiny hydrogen bonds where you're sharing, this small polar molarity, distant ionic bonds between amino acids, and then these hydrophobic and van der Waals reactions. So the other thing, and I probably didn't highlight this enough, things come together if they're both hydrophobic and they're in an aqueous environment, which life usually is, they will stick together with each other. And then that's where the van der Waals can really showcase too. So these surfaces, if these have, nonpolar or, hydrophobic amino acids, they'll want to find other nonpolar hydrophobic amino acids. And that can be how you make a fit between, an antibody and pathogen. Okay. So this is more explorative. Let's go into how we basically try and change this. Sometimes we send in things that will bind to DNA and prevent it from having that good structure. If you prevent that, you can basically rupture DNA, and if you lose DNA this aggressively, you can lose the cell. So, chemotherapy is basically what we use in a lot of cases to treat cancers. The basic principle behind cancer is that you are making too many cells, okay? To make too many cells, you need to have lots of raw materials. One of the raw materials you need to make more cells is nucleotides and DNA, okay? So we send in small, sometimes organic chemicals that will go in and they will bind maybe to nucleotides, maybe they'll bind to the phosphate backbone, but see what they're doing right here. They're basically going to bind and intercalate with this DNA, and now it's stuck and it's being pulled in the wrong direction. And when something comes this way, maybe to say to replicate the DNA because the cell is trying to divide again, given that it's a cancer cell, this gets in the way, this explodes this region of DNA, and the cancer cell dies. The cancer cells are more susceptible to the chemotherapy because they're dividing at a massive rate. But this is going into your other cells. But your other cells don't divide so often, so this damage is, less significant. But it is significant. And that's why we are moving towards things that are more targeted therapies, immunotherapies, things like that. And that's something, I'm excited to get into as well. Chemos don't only have to affect DNA, they can also affect, when the chromosomes are being pulled apart, for example, they can disrupt that process. They can, destroy the chromosome regions that they're pulling apart. The spindle that you see here pulling the chromosomes, they can, basically destroy these microtubules, all kinds of different stuff. You can target a lot of things that are basic cell-required processes, and that's how you can defeat a lot of cancers. Okay, chemo has a kind of a bad name in a rap because of the side effects that are so apparent, but we have defeated a lot of cancers using strategies that surround attacking this cell division process, which is high, much higher in cancer, and attacking DNA because you have to replicate the DNA during the cell replication process. And, you got tons of DNA running around making more of itself. Okay, so I want to go over something very important for oncology and kind of an interesting concept called synthetic lethality. So, most cancer cells lose the ability to detect or fix mutations. Well, some of them do, and that causes more mutations, and that causes, more evolution in the cancer. So, in a normal cell, if you have a big, massive covalent bond break in your DNA, this is a big issue, right? Like we talked about in that non-homologous end joining, that can cause a ton of bad stuff. Usually, what you have are things that can detect the damage or repair it, right? And in a normal cell, you'll probably be okay. Your cells have this happen, and you get exposed to this sometimes. When you have a BRCA mutation, and let's say you lose not one, but both, and sorry, I shouldn't call it BRCA, I should call it BRCA2 in this case. BRCA2 detects these and fixes them. If you've lost both copies, then your repair is not as good. See how it's like okay, but it's not very good. And this is how you start to make cells that not only, let's say they're missing both BRCA2 copies, now they're going to start making new mutations. And those are going to go on to more and more mutations. So this is an advantage the cancer cell has, is the ability to have more genetic change in its populations. Now, if you pair that with something that causes these breaks, so you actually send in chemotherapy that's going to cause way more double-stranded breaks, all of a sudden, the cell has too many breaks. Even a cancer cell can only take so many of these double-stranded breaks, and the cancer cells do not have BRCA. Your normal cells do. Your chemotherapy is actually protected by your normal cells because they still have BRCA active. The cancer's advantage and its strength of losing BRCA is now a weakness. That is synthetic lethality, is turning strength of a disease against itself through therapy in a lot of cases. So in this case, if you induce more double-stranded breaks in a BRCA negative cell, all of a sudden, this cell just starts exploding because there's no way to repair hundreds of these all the time. It's no longer a nice progressive, source of alteration in these cancer populations. It's bad. So on the subject of DNA integrity, you have something called P53 that is called, I think loosely, the angel gene, basically. So what it is, it's this big combination up here of this purple and this blue. Basically, these subunits scan your orange right here, your DNA. See how it's like this is a protein up here and this is DNA. The big TP53 protein is actually going to scan the contour of your DNA. And you see these nucleotides. If you have a G and a C that bind and you have A's and T's that bind, if you mess one of those up, there's kind of going to be a bulge in the DNA. And your P53 is going to meet this and say, okay, this shouldn't be here. Try and edit this out. And like we remember how we saw, you just replace like wholesale, like a whole segment of DNA if it finds a mistake. Or just blow the cell up, because 1 cell out of 10 trillion is fine if we just get rid of it. It's better than if we have to, if cancer develops. So P53 is similar to BRCA in that it is very important for integrity of your cells. So if you lose this, if you lose the angel gene essentially, all these unmitigated damages can start popping up in your DNA, and nobody's there to tell your cells that you have too much damage at that point. So it's similar to BRCA that it's a tumor suppressor gene. So that's something that we'll keep in mind for more advanced stuff. Hooray, chirality. I think this is our last one. Chirality, again, mirror images cannot be superimposed. That means you have two possible versions of the same chemical composition molecule, but as you can see right here, they have a little bit different orientation. So something achiral, you don't have to worry about this. Their mirror images, they're going to be the same. So the thing with nucleic acids and amino acids is that they have the L chiral form. Okay, the D form of these doesn't really exist in nature. I actually don't have a 100% answer why. I can't remember if it's just that I think things just evolved in this nature and never stopped. I guess I can look into that, or you could, because I think it's super interesting, and even though I'm not a chemist, that one form ended up dominating. But in this case, just know that anytime that we're studying it, we are studying the L version of an amino acid or a nucleic acid. And as you can tell, if we would switch these around or anything, it would not fit, basically. We can make proteins and we can make nucleic acids that do have the D conformation. The D form, and they won't work. They won't fit with their chiral opposites. Okay. Almost done. So, last basics, nucleic acids will be affected by pH. When you have a really low pH, you have tons of, for example, if you have a low pH, you have tons of these little hydrogen ions. They will go in and they will pick apart not only your hydrogen bonds between your nucleic acids, but they can definitely damage some of those phosphate chains too, because remember, look at how positive these charges are. They can overwhelm things. Same thing if you have a high pH, which is you have a huge base, and you have tons of these OHs. Remember how reactive OHs are? They'll go in and they'll start binding to everything and ruin your nucleic acids, your proteins, just the same. Temperature similar in that temperature, the higher the temperature, the greater chance you start breaking the bonds. So as you increase temperature, you start to break the weaker bonds, like the H bonds that keep nucleic acids together. But if you increase the temperature enough, you can start, I think, ripping apart the actual covalent bonds, and that actually have to be a pretty extreme temperature, to be honest. I think I'm wrong on that. So kind of a cool application, this should be in blue, by the way, this is just an example. A cool application of this is, let's say we find some Neanderthal DNA. We want to see if there's a region of this DNA that is similar to the human DNA. So over here we have regions of human DNA, and what we're going to do is see if these regions are going to have similar areas as the Neanderthal DNA, because we want to see how similar is the Neanderthal gene to the human gene. And what you can do is, with high enough heat, you can actually break apart those nucleic acids. See how these are broken apart here? Then you expose these broken apart nucleic acids to, similarly, these strands were collected with heat too. You're going to see if they bind, and if they do bind, you're going to be able to, essentially with what these little haptens are, you're going to be able to catch them, and they're also going to light up because they're also going to be carrying a fluorophore with them. So in this case, If you have a region that is similar or conserved between species, you can find out by seeing if you can create a DNA hybrid. Now, that sounds scary. You're not really creating an embryo or anything. You're just seeing if this nucleic acid matches anywhere in the human genome. If it does, you'll get a bunch of these regions hooking in here. So that's kind of the rough way that we used to compare things. Now we can do next-gen sequencing and basically just use the bioinformatics and the computers to compare and see the differences in the regions. This is still a great way to verify things, though, and it's really how we relied on for a lot of stuff. All right, so we will touch on this stuff in a later video, but although this is a molecular biology class and nucleic acids kind of get the special treatment, when we talk about anything that happens in a cell, just like down here especially, antibodies, or even the protein complement system that defends you against antigens, when we talk about something happening, the thing that's doing it is usually going to be a protein. Enzymes, transporters, lots of stuff. So as cool and dynamic as the chemistry of nucleic acids can be, this is where the magic really happens, is when you get to a protein.